Now in chapter 16, we are going to learn more about the acquired immune defense and how antibodies and specific attacks are mounted. So if you recall from chapter 15, the first line defense are those anatomical barriers. Second line defense are things that happen after a breach has occurred, but not specific. So phagocytosis, inflammation, those types of things. Now our adaptive immunity is the third line of defense. So this is that acquired and very specific immunity that occurs after an event like a bacterial infection. So this is going to lead to the production of B and T lymphocytes. We'll be talking a lot about those in this chapter. And those undergo, again, a selective process that specializes them for a specific marker. And it's specifically something on, so it's not necessarily recognizing the whole bacterial cell, but it's recognizing like the LPS, or it's recognizing a specific protein or something that foreign invader has on it. And then just a term here, immunocompetence. This is the ability to react to countless foreign substances. So the way that these lymphocytes are designed is that they could recognize a huge variety of different substances, and only those that get activated, so if they detect that invader, only those that get activated actually go through multiplication and go through producing antibodies or getting rid of virally infected cells. All right, so here just to kind of differentiate between antigen and immunogen. So antigens are also called markers. These are things that can be seen and identified by the immune system. So anything that the immune system recognizes. However, it is possible that an antigen would be recognized but would not provoke an immune response. So then the term immunogen, those are specific types of antigens that do provoke that immune response. So immunogens are always antigens, but not all antigens are immunogens, because it is possible it wouldn't activate the immune system. So again, those immunogens are the ones that activate our B and T cells. So proteins or polysaccharides, they can be exposed, they could be released like a toxin, or they could actually be inside our cells, especially viruses. Any of these can be different types of antigens. So usually our own antigens, though, do not invoke a response. So again, our immune system may recognize them, but it wouldn't create that immune response. And then even some different chemicals can actually activate our immune system. In chapter 15, we talked about PAMPs, those pathogen-associated molecular patterns, as part of our second line defense that gets activated. So again, these stimulate that innate response. So things like inflammation, phagocytosis, again, not a specific reaction. So they're part of our second line defenses. That is different, or those are different than immunogens. So antigens, though, are highly individualized, and they stimulate the adaptive immunity. So PAMPs are part of our innate response, that second line defense. Antigens are going to activate our adaptive immunity. And so the two molecules, though, PAMPs and immunogens, do have a couple similarities. So they are parts of the foreign cells. Again, just the PAMPs are more like patterns like the LPS, peptidoglycan, whereas antigens are more specific. And they do provoke a defensive reaction. It's just whether it's part of the innate or the adaptive immunity. So again, just for this chapter, though, antigen is going to be things that we're talking about here that adaptive immunity is going to recognize. So some of the characteristics unique to the adaptive immunity. So what makes it special? So it is very specific. Again, I keep mentioning that. Innate is general purpose, but adaptive is mounted to one specific part of one specific organism, whether it's a virus or a bacteria. So again, this is showing here, and this is also the term epitope. So it's something that is recognized, one part of this cell, not the whole cell, or one part here of this virus, a capsid protein or something that's very specific. Also, there's a lot of diversity. So there's at least one cell that can recognize any antigen. Again, our adaptive immunity can mount a response to endless types of foreign substances. It can be induced, which just means it can be turned on. So it's not always active. It has to be, kind of hangs out until something is encountered. It can produce clones, so it generates millions of cells that are identical to mount that specificity. And they can exhibit some tolerance, which is where it does not react with self-antigens. And unfortunately, with autoimmune disease, that gets overridden. But typically, the adaptive response has a way, even if it recognizes something, to know whether it's harmful or not. And then finally, the last one that gets highlighted is that the adaptive has memory, just meaning that once we've been exposed to an antigen, then there's rapid mobilization if it's re-encountered. And this is the whole idea behind vaccines, is that if we train the immunity, so if we help it to recognize something as foreign, creates a response, then it's remembered. So if ever the body comes in contact with that again, it can attack it much faster than it did the first time. Whereas with the innate, like phagocytosis and inflammation, fever, that's the same every time. It's not like exposure to something helps fever happen faster or inflammation happen faster. But with adaptive, that memory allows that second exposure to be much faster. So there are four main stages here that happen with that immunological development or the interaction. So we're talking here about the B and T lymphocytes. So first of all, the first stage is just the development and differentiation. So again, the immune system is made to where it could recognize endless numbers of antigens. So just developing those, creating receptors to various antigens. Then if something is encountered, the way that it gets done is by presenting antigens, and that's called presentation of antigens. We'll talk a good bit about that as we go on. And if something foreign, if an antigen is presented to those lymphocytes, then they challenge the B and the T cells. Depending on whether it's bacterial or viral, depends on whether we activate B cells or T cells. So again, we'll come back to that. But ultimately what happens is if B cells get activated, the end result is the production of antibodies. If T cells get activated, then the end result is the destruction of the cell. So this is going to be more viral, whereas the B is going to be more bacterial. And then just kind of going back through these in a bit more detail. So we have this antigen independent development. So this is just the development of B cells and T cells before an antigen is ever encountered. So B cells, that maturation occurs in the bone marrow. So that's kind of easy, B for B cell, B for bone marrow. And then ultimately they go out once they're matured to the lymph nodes, to the spleen, mainly the lymphatic system. Whereas T cells, they're going to develop in the thymus. So again, T cell, thymus, T's go together there. And again, once they develop, then they're going to migrate around in that lymph system as well. Now, once an antigen is actually encountered, so now we are dependent on the antigens. We have lots of different B and T cells hanging out, but until an antigen is encountered, nothing's going to happen. All right, so what's going to happen here? So we have antigen contact. We'll talk about these MHC markers. They're receptors. So they're going to be presented to B cells through that, again, presentation. And so if they get activated, then those B cells are going to mount and have receptors that look just like antibodies that would recognize some part of that antigen. And so if they get activated, those B cells recognize some kind of material. What's going to happen is they're going to be making thousands and thousands of what are called plasma cells, and those plasma cells secrete antibodies. So activation of B cells leads to antibody production. And then we also get some memory cells after that response for that secondary response. If we ever encounter that same antigen, we have a faster way to attack it. Now with our T cells, again, we're still having the antigen. They're going to only recognize antigens that are presented. So we'll talk about that antigen presentation. Whereas with the B cells, those antigens are going to be recognized on the outside of the cell. So again, they have these receptors, they'll recognize them and mount the response. Whereas with the T cells, again, they need that antigen presented on them by a receptor. So they don't necessarily directly recognize the foreign invader. So once it does get activated, once it sees those antigens, activation of those T cells are going to ultimately lead to cytotoxic T cells. And this is where it's going to kill the cells that are infected by viruses. And we still get those memory cells at the end. So B cells lead to antibody production. T cells are going to lead to cytotoxic, so killing of cells infected by viruses. And then ultimately, again, just that antibody production, those antibodies are going to circulate in the blood so that they can encounter the antigen and get rid of it. That's cell mediated. We get some different T cells, but ultimately the cytotoxic T cells are going to destroy cells infected by viruses. And then we also have some regulator and helper T cells we'll talk about as well. Now these MHC molecules are an important part to adaptive immunity, and that stands for major histocompatibility complex. So MHC, this is a set of genes. Genes are pieces of DNA that make protein. So they are making proteins called human cell markers. All right, so we have lots of different ones. These are glycoproteins, meaning it's a protein with a sugar attached. And they are found on all cells except red blood cells. So remember, red blood cells don't have a nucleus. So that's another way to remember they also don't have MHC molecules. Everything else has MHC molecules on them. And those are also called HLA, human leukocyte antigen. So these are going to help recognize self cells versus foreign tissue. So there are three classes of them. MHC class 1 molecules or genes. These are found on all nucleated cells. So again, not red blood cells, but every single cell that we have with a nucleus has an MHC class 1 molecule on it. Now their purpose is to uniquely characterize self. So every cell in our body has these, and that tells our immune cells, hey, this belongs. They have an MHC class 1 molecule, so they must belong in the body. So it's recognition of self. That is their job. That's why all the cells have them. Now the MHC class 2, these are regulatory markers found on only some. So macrophages, dendritic cells, and B cells. They are the only cells in our body that have the class 2 molecules on them. And they are involved in presenting antigens. So these are phagocytic cells. They're breaking down molecules. They take pieces of those broken down viruses or bacteria, and they put them on these MHC class 2 molecules. And that allows for activation of T cells. And then our third class of MHC molecules, they're going to activate that complement system we talked about with the innate immunity as part of the second line defense. So it's a way of activating the complement system. And just looking at these, whether it's class 1 or class 2, again, all cells have the class 1, so they have an antigen binding site. So again, this would helps recognize the cell as being a self cell. Whereas the class 2, those are going to have an antigen binding site, and this presents it. So this antigen binding site would be something that ultimately had come from inside of this cell, some type of foreign substance that's being presented to the T cells. Like, hey, is this bad? Is this okay? What's going on with this molecule? So it has a much different structure. Now the surfaces of our B and T cells are also different because they're looking for different things. So our B cell receptors, they actually look just like an antibody. They have the little Y shape where the antigen binds at the two ends, and then they're embedded at that kind of constant region. And there are lots of different types of antibodies. You'll have to learn if you haven't already, more in anatomy and physiology. We're just general purposing what these antibodies look like. But that's what the receptor on our B cells look like, is an antibody. So they would recognize free-floating antigens. So as a bacterial cell floats across these, that B cell receptor would bind to them and be activated. Now, T cells, though, remember, they only recognize presented antigen. So like a B cell or a dendritic cell would present that antigen on an MHC class II molecule, and then those T cell antigens binding sites would recognize those. And these also have what are called cluster of differentiation. So we'll talk more later about CD4, CD8, and CD3 proteins as well. All right, so this antigen presentation. So first of all, just remembering there are three types of cells that present antigens: dendritic cells, macrophages, and those B cells. These are the ones that are going through phagocytosis or somehow taking in an antigen and presenting it on the MHC class II molecules. So again, the macrophages are going to ingest the pathogens that induces that inflammatory response, but in that process, it is taking in pieces of that antigen and putting it on those class II molecules. Dendritic cells also ingest antigens, migrate to the lymph system, and present it to the T cells. Right, so this antigen challenge and clonal selection. So only the B and T cells that get activated are going to proliferate and keep dividing. And once they get activated, they just make more and more and more of that same exact cell, and those are called clones. So it's a particular lymphocyte. So it's not every B cell in the body that starts activating and reproducing. It's only the one that recognizes the present antigen. So once it gets stimulated, once it's activated, it divides and divides and divides, making millions of the same type of cell, and those are referred to as clones. And again, they're genetically identical, and ultimately we keep some of those around for those memory cells. So we'll start first with how T cells respond to these antigens. And there are three different types of T cells. So we have helper T cells, regulatory T cells, and cytotoxic T cells. Helper T cells activate those macrophages we just talked about, one of the antigen presenting cells. They help with the B cell activation, and they help activate the cytotoxic T cells, hence the name helper T cells. They're helping to activate macrophages, B cells, and T cells. Now, the regulatory T cells, as their name implies, they're more involved in controlling their response, making sure that the correct anti-inflammatory cytokines are released so we don't overactivate the body. It prevents proliferation, so helping to prevent like autoimmune diseases. So it's just kind of keeping the immune system from becoming overactive. And then finally, the cytotoxic T cells we've already mentioned. These are the ones that destroy infected host cells. So if there's a foreign invader like a virus, we just get rid of the cell as a way to kill the virus. Now, B cells are a little more direct. Again, they're activated by the antigen binding directly to it, and it gives rise to those plasma cells that ultimately release antibodies into the tissues and blood, and those antibodies specifically attach to the antigen that destroy them or neutralize them. So we'll talk more about how that works in a minute. Now, those cluster differentiations I mentioned earlier that are on the T cells, so these are just markers that are found on the membrane, and they help with the immune response. More specifically, the CD3 markers, those are on the T cells that help with binding. So they're just helping the receptor bind to the antigen. Then the CD4 and CD8, this is something commonly we hear about with blood work. CD4, cluster of differentiation, those are on the helper T cells, and they bind to the class II molecules. CD8 are on those cytotoxic T cells, and they bind to the class I molecules. So what that means is if we have a high or an increased amount of CD4 markers in the body, so whenever they do blood work, if there's a lot of CD4, then that indicates that there's a bacterial infection because it's the helper T cells that are getting activated. Whereas if we have a lot of CD8, if that is elevated, that means we have a lot of proliferation of cytotoxic T cells, indicating it's a viral infection. Right, nice little summary table here just to differentiate between our B and T cells. We already mentioned where they mature. So bone marrow is where B cells mature. Thymus is where the T cells. The surface receptors or markers on each one. So the B cell actually looks like an antibody, is actually going to be the receptor, whereas T cells have distinct T cell receptors that recognize those presented antigens. We don't have a lot of B cells in the body until we get an infection and they proliferate and start dividing, whereas there are lots of different T cells circulating in our immune response. Again, the different receptors are B cell and T cell receptors. I'm not concerned that you know necessarily these locations specifically, but again, the difference, B cells directly recognize the antigen. It doesn't have to be presented, whereas T cells only recognize presented antigen. So they wouldn't recognize a bacterial cell, but they would recognize something presented, a tiny piece of it presented on one of those class II molecules. And then the result of activation, again, those plasma cells that lead to, antibodies. Whereas the T cells, we have different types of T cells, but ultimately those cytotoxic are going to help kill abnormal cells. They can recognize different cytokines in our immunity to help with that immune response. Diversity that we see with our immune system is due to those different types of antigen receptors that recognize unique antigens. Okay, so those B cells are going to kind of hang out in the bone marrow until they get activated. And then the T cells, again, they migrate to the thymus. But each one of them, each different B or T cell has a unique receptor. So it's not every B cell in our body pretty much has a different receptor to recognize a different antigen. Same thing with the T cell. So they migrate to those secondary lymphoid tissues, like again, the spleen or lymph nodes, but only if they get activated do they make more and more of themselves. And that allows that receptor to be more active to recognize the antigen that is present at the time. But again, for the majority of these B and T cells, they recognize a single unique antigen. So there are millions of different B cells because each one has a different B cell receptor. Same thing with our T cells. So if we look at these stages, so again, our B cells here in the bone marrow where we have these immature lymphocytes. So the B cells are going to stay there in the bone marrow to mature. T cells, they'll go to the thymus to mature. And what that maturing means is just whenever the cells are originally made, they don't have any receptor. Okay, so it's an immature lymphocyte. So that's made in the bone marrow. Again, B cells stay there, T cells go to the thymus. And then as they mature, the difference is that's when that receptor gets made. So again, our immunoglobulins are just antibodies, but each B cell is going to have a different receptor, which would recognize a different antigen. So this B cell is going to recognize one antigen. Another B cell still has those receptors, but it recognizes something totally different. And then another B cell, the same thing. So they have receptors, but each receptor recognizes something different. Same thing with the T cells. So they all have T cell receptors to recognize those presented antigens, but they're all going to recognize different types of antigens. And then ultimately, once they are mature and they have their receptors, then they go to those secondary lymphoid tissues like lymph nodes or the spleen to actually encounter those antigens. So now the role here of this antigen processing and presentation, because this is, again, really important part to this adaptive immunity. Again, mentioning again, so it shows the importance of this. There are 3 types of antigen presenting cells: macrophages, B cells, dendritic cells. And what it means to present it is that the cell literally grabs that antigen carrying microbe, so say a virus or a bacteria, it ingests that, it chomps it up into those little lysosomes. So we think kind of like, again, phagocytosis. So it's bringing it in, chopping it up into little pieces, but instead of just releasing those pieces like we had seen with phagocytosis, now what happens is they take pieces of that antigen that was broken down in the lysosomes and they put it back out on the MHC class 1 or 2 markers. So again, MHC1, those help recognize self, so that even our self cells are doing this and are like, oh no, this belongs here. These little antigens, they're part of our normal cells. Don't be activated. They're normal. Whereas if it's something foreign, it's put on that MHC class 2, like a piece from a virus or a piece from a bacteria, so that those immune cells can recognize it. And this is just showing that process. So here we have this antigen presenting cell. Again, macrophages, dendritic cells, and B cells all do this. So they recognize or see this microbe, they bring it in, they chomp it up by those lysosomes, just like we saw with phagocytosis. The difference, though, is that the little pieces that are broken down, instead of just releasing them out of the cell to get rid of them, they take some of those pieces and they put them on those MHC molecules. So if it's a self protein or something that belongs there, it would put it on a class 1 molecule. If it is something foreign, it would put it on the class 2 molecule. So then what happens here with our T cell receptors is they're going to recognize the MHC class molecule and the antigen on it. So these T cells would never recognize something like the whole microbe or the microbe free floating, only something that had been chomped up and put out on these MHC molecules. And then also those CD8 or CD4 molecules that help with that binding process. And this is just showing kind of enlarged here. So we have our helper T cell. So this T cell receptor here is recognizing what is being presented by one of these antigen presenting cells. So it recognizes the MHC class molecule and the antigen that is presented on it. All right, so looking at this T cell activation, because we have a few different types of T cells. So what's going to happen here is we have, again, our antigen presenting cell. So our antigen presenting cell, B cell, dendritic cell, macrophage. It has its presented antigen on here. In this case, it's a class 2, which means it's something foreign. And so here we have this CD4 cluster differentiation. Again, that came from something free floating, so it indicates it's a bacterial infection or something outside of the cell. So once it gets activated, this T cell is now going to do a few things. One, it's going to create memory T cells that recognize that antigen. It's also, though, going to produce helper T cells. And these helper T cells present that, and that's going to help activate the B cells. And then activation of those B cells leads to the formation of plasma cells that release antibodies. And then the antibodies have that antigen binding site that would then go and bind to the bacteria. And then we get some memory cells left over from that. And also there are some regulatory B cells that help control the immune response once it's over. Now we also have some other helper T cells. Some of them help produce tumor necrosis factor. and that stimulates macrophages. They're involved in helping to get rid of tumors, actually, and prevent cancer. Unfortunately, it doesn't always work, but that's their job. And then some of the other helper T cells are involved in things like allergies, inflammation, and just those regulatory T cells that help inhibit the immune system, so that especially once the reaction is over and the antigen is gone, now we kind of bring everything back to normal. So again, we have lots of different T cells here, but those involved with the CD4, they're recognizing antigens that came from somewhere outside of the cell. Once they get activated, they help activate those B cells to create the antibodies. The T cells that have the CD8 markers on them, these are ones that are recognizing potentially virally infected cells. So here we have that MHC class 1. Again, that's presenting stuff from inside of the cell. So it's not an, it's something that came from inside. So we still have our antigen presenting cell. It is having those MHC class 1 molecules, something that came from inside of the cell. And if that CD8 cluster of differentiation and that antigen binding site of its receptor gets activated, that indicates that there's a viral infection. So that's going to activate that T cell, and it's going to lead to the production of cytotoxic, so TC cells. And they are going to recognize an infected self cell. And if that gets activated, then it's going to destroy that host cell. So this cytotoxic T cell releases enzymes and molecules that ultimately kill the infected host cell as a way of getting rid of that virus or that infection. Okay, so again, our CD4 T cells, they're involved in activating B cells to produce antibodies, so more bacterial infection. CD8 T cells, they're involved in recognizing those self cells, and if there's something foreign, like a viral protein that ends up on a self cell, then they get activated and they destroy that infected cell. That's using the cytotoxic T cells. Now our B cells get activated in a different way. All right, they don't need that antigen presentation originally. They bind directly to the antigen. So again, here's our microbe. Those B cells are going to recognize something on it. They're going to attach to it, so it literally binds to the antigen. Remember, B cells, though, they're one of the antigen presenting. So once it binds, it's going to internalize or phagocytose that antigen. It's going to chomp it down, chop it up into little pieces, and then it's going to present some of those pieces on those MHC class 2 molecules. And then what it's going to do, once it processes and presents that, it is going to wait for helper T cells to essentially confirm that that is foreign. So this helps, again, regulate so we don't get those autoimmunes. We're not overactivating the immune system. So the B cell binds directly to the antigen, brings it in, chomps it up, puts it on those MHC class 2 molecules, and then it waits for confirmation from those helper T cells. So if the helper T cell comes along and goes, oh, yep, that doesn't belong, again, using those CD4 markers, also the CD3 help it to attach as well. But if we get activation and it says, the helper T says, yep, that's foreign, then that is going to activate that B cell. And activation of the B cell then leads to that antibody production. And that's what we see here at the bottom of that figure. So once that B cell gets activated, again, confirmation from that helper T cell, now it produces plasma cells that make lots and lots and lots of antibodies, but we also get those memory cells and regulatory B cells left over too to create those secondary responses.